So here is an example, as you can see, detection of BCR ABL1. So these are the two names of particular uh, things, the genes, right? So BCR is a gene. So we have to talk about the gene rearrangements and how they lead to particular disease condition and how you can use fish assay for detecting it. So this is an example of BCR and ABL1. Now here, what is happening? Let's understand that and then how the technique is utilized to explore its consequences. So what is happening is reciprocal translocation. That means a part of one gene is transferred to another gene and there it gets ligated. So cutting of the two genes in the two different chromosomes maybe is happening, and the gene segment is moving from one gene to another and getting ligated over there, right? So that is translocation. It is happening between chromosome number 22 and chromosome number 9, and that results in the condition which is referred to as Philadelphia chromosome. So chromosome number 22, chromosome number 9, they will undergo breaking and breakpoint, and rearrangement of the sequence will take place, and that will result in a condition which is called as myelogenous leukemia, blood cancer. Okay, and this modified gene that has resulted from reciprocal translocation between two chromosomes, that is chromosome number 22 and chromosome number 9, is referred to as Philadelphia chromosome. Now, the chromosome breakpoint on chromosome number 22, for this translocation, it is located within the BCR gene. So, chromosome क्या है? 22, उसमें जीन कौन सा है? BCR. So the translocation produces a fusion protein. Naturally, when a gene is cut, right? I will go to the next slide so it will be little more visible to you what I'm talking. So this is chromosome number 9, this is chromosome number 22. Here you have the BCR gene, and on chromosome number 9 you have the ABL gene. They have their own functions, right? Regulatory functions, and enzymic functions are many known to or assigned to this BCR gene. I'm not going to go into their uh, uh, details of the role, but the tyrosine protein kinase, which is a very vital key enzyme, and which is going to be involved in many of the processes related to cell growth and survival, cytoskeleton remodeling, etc. Is this particular region, right? So you can imagine if a region which is involved in remodeling in the response to extracellular stimulus, cell motility, adhesion, receptor endocytosis, autophagy, DNA damage response, apoptosis, इतने सारे functions में, so how key role this particular molecule has, and if it gets rearranged, naturally it is going to cause an havoc, right? So this particular sequence is very important because it is resulting into expression of a product that is tyrosine protein kinase, and which has got very much what we say important role. All right. Now what is happening is chromosome number 9 with ABL gene and 22 is with BCR gene. Now what has happened, as the picture clear cut shows to you, that the cut at ABL gene and cut at BCR gene has happened, and this is now the rearranged chromosome. You can say as a change chromosome number 9, and again BCR also is changed. Now this changed BCR region, gene is referred to as Philadelphia chromosome, which is having BCR region as well as ABL gene. So these are two genes which are got fused in chromosome number 22, and this fusion gene is going to produce fusion protein, right? So this is what we go back to the previous slide again. So here this fusion protein will be expressed now because the gene has got rearranged. As a result, the function is going to get altered, right? The unregulated tyrosine kinase activity of ABL1 contributes to immortality of leukemic cells. So immortality, that means the cells are going to continuously divide. That is what we call it as cancer, right? So the BCR protein has serine threonine kinase activity and is a GTPase activating protein for P21 RAC and other kinases. So these are the names of different genes and their sequences, genes and their products, right? So of course, these details you can get to know if you go into details related to the particular disease, that is leukemia here in this case. That is not our concern right now. Our concern is the rearrangement of genes 22 and 9, it results in a fusion protein, sorry, fusion chromosome, and that fusion chromosome is going to have effect, that is immortality of leukemic cells. All right. Now, uh, here, this is detected using the dual fusion probes, right? Dual fusion probes. Now, what is used? A metaphase tissue is required. Metaphase, because it is, the duplication of the chromosomal sets has happened, and the cell is in the phase at metaphase, the rearrangement of chromosomal sets at the center plate that happens, that. So that is the stage of the cells that is selected. Fish technique is done, and what you are using is dual fusion probes. So what is dual fusion probe? That is, you are going to have two different probes. One is green labeled and one is red labeled, for example, as you are seeing in this particular photograph also. So green fluorescent probe is designed to find the breakthrough region of the BCR gene, because there are two genes breaking at 2 points. So specific for BCR and specific for ABL, you have two fluorescent probes. That is why it is referred to as dual fusion probe. So you can localize the presence of these fusion um, uh, uh, regions. If it is only green, that means it is intact, say BCR, and if it is only red, that means it is intact ABL. But green and red together, right? So green and red together in this particular region, you can see, that means it is fused. So the sequence जो है, the probe is having green fluorescent probe, it is binding with one gene, and red fluorescent probe, it is binding specifically with another gene. If you see a fluorescence of both the colors next to next, that means fusion has occurred in this particular region. So you can find out how many cells are having such altered sequences, right? This is what is been tapped here. So you can look for the chromosomal rearrangements that have happened and have led to development of a particular disease. And a very good example here is that of what we say, rearrangement of BCR-ABL genes in chromosome number 22 and chromosome number 9, which results in the leukemia, right? So an immortality of leukemic cell is on account of the fusion protein that is produced by fusion of BCR and ABL1. Right. Now here are some more details about it. This is what is the what we say, website, like you have the databases available on such websites that talk about the particular proteins, disease conditions, and everything you have. Swissprot. Last year we have learned about it. So this is UniProt, right? So it is telling you about the protein, which is P00519 ABL1 underscore human. So source is human. This is the gene, right? This is the code number given. What is that protein involved in? It is tyrosine protein kinase, right? What is the gene? ABL1 gene. And what is the status? Uniprot KB reviewed Swissprot. That is the database name. We did not worry about it. So obtained from Homo sapiens. What are the amino acid composition? There are about 1130 in the sequence. So protein existence, yes, evidence at protein level, and annotation score. 100%, you can say 5 by 5, that is complete annotation is available. So this is how data about these gene sequences are available on internet also, in the protein databases also, nucleic acid databases also. So this fish technique can be used to find out the presence of such rearranged chromosomes. So breaking reunion and formation of an altered sequence. In this case, Philadelphia chromosome, we have taken an example. There are many such conditions where there are rearrangements, where there are multiple copies, like trisomy in the previous example we have seen, right? So they can be very well identified when you are talking about this particular technique, right? Now, one more thing I wanted to share with you here is the applications of this particular technique. We have seen a few applications in the beginning, right? So this is what we had ended our previous lecture, and this is what, no, not this. Yeah. So the steps, it is just take a snapshot of this we have discussed about it, right? If you want, I can repeat the steps like protocol, fixation, permeabilization, hybridization, washing, and visualization or detection of the uh, stained or fluorescently labeled samples. That is how we go for. Right now, when it comes to first step, that is fixation step. Fixation is going to allow preserving nucleic acid integrity and cell morphology, and this is what all we have talked about, right? It is just for writing purpose if you need any extra information. So understanding the internalization mechanism is crucial because you have to alter the membrane integrity. The DNA has to enter into it, or the RNA probe has to enter into it. Then the fixatives which are applied, such as paraformaldehyde or ethanol, can be used to permeabilize the cell membranes. And that of course you have different types of fixatives, and depending upon what is your target, the fixative also is going to be very important, because what may work for animal cell or human cell may not work for gram positive cell or gram negative bacterial cell, right? So different types of fixatives may be applied, and pre-treatment of course may be required, as in case of gram positive bacteria. So permeabilization agents may be like enzymes or solvents, detergents also are used, and even hydrochloric acid is also referred. So fixation step is fix is, as in to fix on the slide, that is how we get it. But another thing is permeabilization is allowed so that probe enters, right? So this is the first step. Second is hybridization step. Then third is after hybridization, what is important is uh, detection of the hybridization product. So the fluorescent probes, when you say, see, if there are very less number of bacteria in a given sample, so fluorescence will be less, right? So what you need to have is a good intense signal has to get developed as a result of hybridization, right? So hybridization product detection is going to depend on the fluorescence that is generated by binding of the fluorescent probe to the target, and if the target sequences are less in number, the fluorescence will be very less. So signal detection should be good. So uh, distribution of one or more types of microorganisms in a complex matrix also is going to be another challenge, right? So as we have seen in previous example of Philadelphia chromosome, green and red you could see, but if there are different types of bacteria in given particular sample, then it will be a challenge. We will come to that example little later, but I'm referring to this as a point, that is uh, uh, visualization or detection when it comes to, so it will be one of the challenge. Then how to deal with it, that we will talk. So uh, you are going to uh, visualize these fluorescence signals, and the efficiency when you talk about these system is going to be a microscope which is going to allow the process without loss of any signals. There shouldn't be any noise. So these are the technological things where quenching can happen, because we are talking about fluorescence, right, phenomena. So when the light is generated, if there are certain components may be part of fixative or maybe permeabilization agent, which is going to absorb that fluorescence that is generated, then the signal will be poor or weak, and then detection will be difficult. So this quenching should be avoided when you are optimizing a protocol for permeabilization or fixation of the tissue. That is the care to be taken. Now the point is, when you are talking about optimization for different variables in fish process, you have to go by trial and error method, changing one variable at a time. Changing one variable at a time is one concept, that is what you use in fermentation experiments also to optimize the media, right? But advanced uh, and most acceptable method is RSM. What is that? Response surface methodology, in which you have to go with multivariate statistic technique. That is the most important crucial factors which are going to detect or have effect on the product that are to be identified, and their combination and permutations are to be tried. So it is a kind of software, these are available. RSM softwares are available, like yeah, you have statistics softwares. Similarly, you have software for response surface methodology, For example, design expert is one of those, and you can give the parameters, their range of working, and using that data it will generate the sets of experiments with varying combinations of the important parameters, and the set can be developed, and you have to select from that and do only that experiment instead of running making 10 to 100 combinations and seeing which is giving you the best product. Instead of that, you have to identify the best factor which is most influential, put their range of working, and ask the computer to generate the set of experiments, and the experiment which is showing better yield on computer analysis, that you are going to try in the laboratory. That is what is the crux of RSM. It could be another lecture on RSM, so I'm not going to go into details, but it is just to make the screening procedure most efficient and optimum. So here also, for optimization of fish variables, you can take help of RSM, and the softwares like Statis or Design Expert, they are available on the market. But yes, it is very crucial. Now, okay, ionic strength, temperature, buffer concentrations, the detergent concentration, or the permeabilization agents which are using the concentration of those, all these things are going to be very crucial and need to be optimized. All right. Now, if I have something like say a sample, uh, maybe tissue that we have taken an example of, particular related to disease or particular bacterial species that you want to identify. These two examples we have seen. But is this the limitation of the particular technique? No, you can use it for various purposes. And here is an example like ecological, phylogenetic, diagnostic, environmental studies. Right. So right from water to soil and human tissues to fermentation, some broth, anything could be your sample. Or maybe when you are doing plant tissue culture or animal tissue culture, in that presence of any contaminating organism which is not detectable by any other means can be identified using this fish tool. Right. So for example, here last application, if you see, this particular bacteria was suggested to be involved in strawberry flavor synthesis, biosynthesis by fermentation methods, right? And this bacteria was a contaminant, so that can be very well identified. Or you can identify different disease causing bacteria or viruses which are there in marine organisms. So if you are doing aquaculture and your animals, I mean the reared organisms, they are getting killed, and you don't get to know what disease it is, you can use this technique to find out. You can find out the symbiotic relationship between different bacteria and the host cell, and the bacteria, between two bacteria or bacteria and plant or bacteria and animal cell, between two bacterial cells also. So these are the different you can say the applications, that is existence of bacteria intracellularly in any of the host cells, right? Maybe a parasite, maybe a symbiont, that can be detected. So these are the different applications. But when it comes to say ecological samples like say soil sample, right? Now what can be done? Now here is the example which I have taken, that is soil microflora or ocean habitat. If you are talking about, right, huge number of microorganisms are going to be there, thousands of target molecules will be present. So how to go about it, right? So here is a case, microflora from soil or ocean habitat to obtain a detectable fish signal. So monolabeled frozen oligonucleotide probe, a few thousand rRNA target molecules are required. You getting the point? So this is the complexity level when you are talking about a particular technique. So you should have a detectable signal, right? So monolabeled frozen oligonucleotide probe, when you are using, few thousand RNA target molecules are required to give you good signal of your interest. There should not be any noise, right? So the standard procedure of fish, if you perform, it will be limited to identification of microbe with high ribosome content only. Or maybe if you are talking about RNA, then ribosome. For if it is DNA, then multiple copies of that particular sequence or particular bacteria in the particular colony. So of different types of bacteria, if they are there, so signal will be very weak and very poor. And there you need to have multiple copies, then and then only you can get a good signal. So these are the limitations of normal fish. So when it comes to challenges like this, you have advanced methods of fish technology, which you can say the variations or of the technique, right? For example, in this case, to detection of fish stain cells in soil, mostly affected by autofluorescent soil particles. So weak signal is one, autofluorescent soil particles, autofluorescent pigment materials, autofluorescent proteins. So many things are there, right? So that fluorescence will interfere with the fluorescence of the probe. So that is another challenge. Then oligonucleotide probes labeled with the enzyme horseradish peroxidase. So this is what you have used in ELISA also probably, right? So autofluorescence probes, if you are talking about autofluorescent sample components, if you are talking about, then fluorescent probes will be of no use or have limited application. So can you change? So in this case, and they have suggested use of enzyme HRP, right? So this is one thing, but the improved methods, when it comes to, there are different improved methods. For example, it is card fish, CARDFISH. What is it? Card is to catalyzed reporter deposition. So optimize permeabilization of target cell, leading to an increase of detection rate up to 86%. So variation of fish is card fish, right? So when it comes to detection of biofilms, detection of marine environments, or the Bethnic detritus, complex organic materials, that time you can take help of card fish. So are there any such variations? So I'm going to talk about the variations here. uh, Fine. So here are some of the variations, like other than cardfish. Here is marfish, it is nano SIMS fish, it is BONCAT, born catfish, right? So here are the schematic representations and what is the variation, that is very well beautifully shown. Now in this case, if you compare isotope dependent methods and non-isotope dependent methods, so it is about the labels that are used. So radio labels, when you are used, you are saying isotope dependent methods and they are referred to as MAR fish. And when you are talking about non-isotope dependent method, so inclusion of non-canonical amino acids, AHA like, right? So they are referred to as born catfish, right? Base of the rest of the technique is going to be similar, right? But if it is the radioactive label, then autoradiography will be required. But if it is fluorescence, then fluorescence microscope of flow cytometry will be required. That is how the variations are there in detecting systems and the probes which are going to work. So here, if you look about MAR FISH, what it is, you have to incubate with radio label substrate. Then you have to go for autoradiography, right? And then hybridize cells with a fluorescently labeled tRNA targeted probes, and then you are going for analysis. Here it is beam of primary ions is scanned across the sample surface, then hybridize the cells with fluorescently labeled RNA targeted probes, and then you are going for analysis. And when it is about non-isotope dependent born catfish method, you have to incorporate the specific amino acid, that is AHA, into it. This is the structure for AHA. And then the hybridized cells with a fluorescently labeled tRNA targeted probes, they are taken for analysis. So this is about incorporation of certain additional components, may it be a radiolabel compound, and then you are going for hybridization of the targeted probe to examine, as in case of MAR, right? Or it is nano SIMS fish. When you are talking, the hybridized cells are analyzed by CLSM, and then analysis of metabolic activity is obtained by this instrument. And when it is to BONCAT, I'll just, yeah, BONCAT approach, then the you are going to conduct the BONCAT assay, that is in vivo incorporation of the non-canonical amino acid AHA, that is azido homoalanine. And what you are seeing, the green cells in the figure right here. So the they are combined with fluorescently labeled RNA targeted probe, and the fish assay is carried out. So this is how the variations are. Of course, uh, they are not there in the syllabus, but yes, we you need to know okay, how the different advancements in the technique are and what is the purpose, right? So Just last 2 minutes and then we will conclude. Yeah, here are the important improvements which are going to increase the accuracy of fish methodology. So classy fish and HIPR fish, that is another thing. So these are multiplexed. So you in next generation sequencing or yesterday microarrays we talked. So they are referred to as multiplex. Multiple analysis at a time are happening. So this is the version that is classy fish and HIPR fish. So that is advancement. Then bone catfish, just now I talked to you about for the study of microbial activity in situ. So it's not only about detecting the presence of a particular sequence, but it is about tapping the activity, right? Then SIP fish and Raman ACM, Nano SIMS. What is Raman spectra? ACM is to ACM scanning electron microscopy. So many techniques are put together so that you can do in situ identification and simultaneously analyze the structure, physiology, and metabolic activity of the cell. Everything in one go. So such very advanced instrumentations are there. So basic is FIS is fish, but you are putting it together in combination with spectroscopy and electron microscopy and so on and so forth, so that entire analysis, in situ identification, and structure, physiology, metabolic activity of the cell, all that can be observed. Another is parsec fish. The sec word is telling you it is about sequence. So expression of various genes, sequence, that is what is done with this particular technique. You can understand interaction among microorganism metabolites, genes in the biofilm communities, and their expressions, their effects. And from ecological point of view, also you can study how a particular biofilm is architectured or designed or developed, how it is working, and what are the crucial molecules, what are the quorum sensing molecules and their effects, so on and so forth. So fish based techniques coupled with various cutting edge technologies like machine learning also are there. So n number of advancements are coming in this particular area, and it is just not about DNA hybridization and finding out a particular gene in a chromosome in the tissue or host cells, but it is much more than that and has applications in all diverse fields, including physiology, pathology, diagnostics, genomics, to biofilms and soil, that is ecological analysis. So this is what the summary of fish method is, and this is the first slide that we have started with.